In this video, I'm going to talk about in detail translation. So before this video, I made a transcription video. So normally a translation is a process that happens after transcription. So make sure to check out the transcription part first. Basically, I'm using an analogy, the Bob the Builder analogy, which you can see is this process here. Which matches exactly the real process which happens, which I showed right under here. So essentially what happens is Bob the Builder really wants to build a rocket, but he needs three things to be able to build this rocket. He needs information, and he needs information, which is the book where the information is. But the issue is when he goes to get that book in his library of his house, he finds a problem. His dad is working and he's not allowed to go in, so the door is locked. So what he does is he asks his dad to just pass him the book real quick, but he can't because the door is locked and the book does not fit under there. But what he does is he's smart. He knows that he only needs one page of that book, because this book explains how to build everything in the world, but he only needs the page on how to build a rocket. So his dad, what he does, so he tells his dad to go photocopy that page with the photocopier and just pass him the page under the door, because now it will fit, right? Because now the book doesn't fit, but a single page will. So that process, the book, photocopier makes a single page. That whole process is transcription. So this I explained in the previous video. So in real life sense, so at DNA, the book represents DNA. DNA is massive, it's got all the information. And then the photocopier is RNA polymerase. So RNA polymerase is like an enzyme that is basically going to do what the photocopier did. It's going to copy a specific section of the DNA to give you a small section copied of this DNA will give you mRNA, messenger RNA. So if you want to be clear on how exactly this process happens, I went into detail that you need to know, all the details that you need to know to do well on your test. Go check out transcription. But now we're going to go into detail on translation. So now that we have the instructions, Bob the Builder needs more things. Just leaving the paper on the floor there won't build a rocket for him, right? So what he needs is materials and his toolkit. He needs two other things. So these three things combined and his intelligence will give you this rocket, right? So same process in the real body. So we have this instructions, this specific part that is going to teach us or that is information on how to build this final thing that your cell needs, which is called a protein. So your cell is trying to build a protein. Proteins are so useful because they allow for everything to happen in your body. Your body needs proteins, and it will send this protein somewhere where it is going to be used. So in the cell sense, the materials is tRNA, transfer RNA. Transfer RNA is legit the thing that is going to be the materials. And ribosome is going to be like the toolkit. It's going to be what is going to it's like Bob the Builder's toolkit. So you can see it exactly matches. Now let's go into detail on how the specific information that we copied from the DNA is going to be turned into protein using just these three things: mRNA, tRNA, and ribosome. So bear in mind, the transcription happens in his library, which is in the cell, represents the nucleus, right? Because we know that DNA lies within the nucleus of the cell. Now, after that, so this transcription is in the nucleus. After that, he's going to go to his garage, because that's where we build stuff, right? In the garage. Now, the garage in the cell represents the cytoplasm. The cytoplasm is basically what is surrounding the DNA. So everything outside the DNA, outside the nucleus. The nucleus is like a small circle in the cell. So the cytoplasm, where he's going to go and build this, is outside the nucleus, which is in the cytoplasm. So you can see here, he's got everything he needs. The paper, toolkit, materials. Now let's go see that process. So remember, not going to cover transcription. Go and watch that video and you'll see what I was doing there. But now translation. So here we got it. So this is his instructions. Now he has his instructions. This is how it's going to look like in the cell. It's going to be a small section of this DNA that is coded by letters, right? Now he needs two other things, like I said, the ribosome and tRNA. How does the ribosome look like? So a ribosome looks, don't worry, the shape doesn't matter. Just know it's kind of like this. It's got like two parts to it. But you don't need to know that. Just know it looks like this kind of shape. And then there's tRNA. So this will be the machines. Now the materials. Here we got tRNA. Just this part. tRNA. Okay? So don't worry about all these things I labeled here. I'm gonna go into detail on that just now. Just bear in mind that we have the three things that we need. The mRNA, the tRNA, the ribosome. The instructions, the materials, and the machinery. Just so you know, mRNA. Yeah, this is just for you to bear in mind. This is like the tools. That's like his paper. Don't need to worry about that. So here's the process. Just delete these don't matter. So yeah, you can see the resemblance. Now, let's explain a bit more what's going on now. So your mRNA, so the instructions, how on earth is your body gonna know which material to bring so that you can build this rocket or this protein? How is it gonna know? Well, the key lies in these letters. Just like English, when we read a book, we know what it means, right? We know what the words mean. Now, in your DNA, these letters also mean something. They're not just there. So what specifically happens is, you know how in English we have different lengths of words, right? A different length means a different word. Some words are 10 letters, some are 2 letters, you know. But in DNA, it's different. Each word is only 3 letters. Okay? So there are no 4-letter words, no 2-letter words, they're all 3 letters. And in English, we call them words, right? Now, in DNA, we call them codon. So, in DNA, the words are codons. So, this is one codon, and then the next 3 letters will be another codon, the next 3 letters will be another codon, next 3 letters, another codon. So, it's made up of words called codons, okay? And they sound like code, right? They're coding for something. Just like words in English, they code for something, they mean something. Now, each of these words are going to code for an amino acid. An amino acid is like a material. And this amino acid, this is also an amino acid, is going to be carried by tRNA. So tRNA is the thing that is going to carry the materials. It's going to bring the materials that are needed to build the protein. Okay? So this one mRNA codes for the materials using codons, and tRNA, which has the ant-codons are pretty much going to match the codon. So this codon is going to code for a specific ant-codon. The ant-codon will be on a tRNA, which will have the specific amino acid for that codon. So AUG will code for a specific tRNA with a specific amino acid. They won't randomly match, right? They won't randomly match everywhere. One codon will match with a specific ant-codon, which will have a specific amino acid on it. And once that amino acid is brought, that's going to be the beginning of building this rocket or this protein. As you can see in this diagram, one of these circles is amino acid. After putting many of them together, according to your code, you will have a protein that will mean something in your body, have a specific function. Okay, we'll go into the process of how what I just explained happens. So just bear in mind the amino acids are represented by little circles. So here we go. So here we have, so this is in your cytoplasm, remember? So here we have a ribosome, right? The 2 gray parts. And here we have mRNA. This is your instructions. So the instructions is going to insert into the ribosome. Okay? So the ribosome is like the tool. It's gonna insert, the mRNA is gonna insert into the ribosome. That's kind of like gonna hold it so it can use it. Now the tRNA, remember I said, has the materials, but the ribosome is gonna like read. It's gonna read these instructions, and it's gonna hold this mRNA, and these codons are gonna code for specific tRNA that's gonna have the specific amino acid that it needs. So let's look. So step 1, we will need a codon that's gonna start the process. Okay, we need like an on switch. So bear in mind that there's always a start codon, and that will make the whole process begin, right? So in the body, there is only one start codon. So a specific 3 letters are gonna mean start. So AUG always means start, always. AUG, and I remember it thinking of just August. It's just a quick way to remember. It doesn't mean anything, but AUG is short for August. So they always start in AUG. So AUG is going to code for a tRNA that's going to cause the process of translation to begin, to start. So how does this specifically happen? Now, how it does that is by complementary base pairing. Remember, A always binds with a specific other letter, right? So what are the letters of DNA? This is important. So the letters of DNA are A, T, G, and C. A always binds with T, right? And this is easy to remember because A and T are always together. A and T are always together. So in DNA, A will always bind to T, G will always bind to C. That is called complementary base pairing. These letters complement each other. They don't complement any other letter. G and C will always be together, and A and T will always be together. But there's one difference with RNA. RNA has almost all the same letters except one. Instead of having a T, it has a U. So every time that you are going to try and put these tRNAs together with the codons and anticodons together, you have to bear in mind every time there was going to be a T for DNA, you will instead have to put a U. Let me show you what I mean by this. So this is mRNA. So this is already an this is already an RNA, which means that it won't have any T's. Every time there was a T, it will have a U, a uracil, a U. See, there's no T's here. That's how you know it's an RNA. This is messenger RNA. It's the message. Now, A is going to bind. Normally, it binds to T, right? But we know these are just also RNAs. They're tRNAs. So they won't have T's. Instead, they will have what? Every time that there was supposed to be a T, in RNA there will be a U. So we know the first letter that it's going to code for is U. So now let's look. Where's the first? This is U. This is U. Oh, so it's going to be one of these two that it's going to call for. The first 3 letters. Remember, we're focusing on the first 3 letters, the codon. So now next one, U. So normally in DNA, the U would be a T, right? But this is RNA, so it's a U. But in DNA, suppose this was a T, what would it bind to? Remember, A and T are always together. So if this was a T, it would bind to A. But RNA, this is a U, so the U will bind to an A. So it's going to be still either of these two. So the last one will make the difference between these tRNAs that are floating around with the material. So G always binds with C, right? G always binds with C. So it's going to be this one. So what's going to happen is this tRNA is going to come and do complementary base pairing with the mRNA. And that's going to, now the process is switched on. Next, we have another 3, right? Another codon. So it's going to call for another one. We know this is going to be this one. You can look at this by yourself, but I know that it's this one. I don't want to waste too much time explaining it again. But remember that it's going to be this one. So it's going to come in. And bear in mind, at any moment, there are only allowed to be 2 tRNAs in a ribosome, never 3. And you can see this is not possible because even if another one came, it wouldn't fit in. It would be off the ribosome. So at any moment, there are only allowed to be 2. So does this make sense? A is normally in DNA would bind with T, right? But we know this is RNA, so there's no T, there's a U, so this works. U in DNA is normally a T, and T normally binds with A, so this works. C always binds with G, so this is correct. Now what's going to happen? Now this materials need to be linked together, because you know when you build a rocket you need to link all the materials together to build this final product, right? So this material is going to now transfer to this one, like this. So it's going to lose this and transfer it to here. And this one will now become this. Do you understand? So now we transfer that one to here. Now there's two linked here. Now the ribosome will move, because this one, it now finished the first one, but now it wants to bind another one, but there's no space. So the ribosome's got to shift a little bit. Okay, it's got to shift a little bit to be able to now, and now this one will not fit anymore, so it will fall off and go away. Because now we have a space for another one. Now this one is UCG. UCG codes, this codon codes for what? Codes for which of these anticodons? So remember, in DNA, there is no U, there's normally a T, and T codes for A, right? So both of these have the beginning letter A. Now the next one is C. C always goes with G, so we know it's going to be this one, because this one has the G. Now, same thing will repeat. Now, these two amino acids here will transfer to this tRNA's amino acid and form an even longer chain. Okay, it's going to transfer it all the way here and going to form this. And now again, the ribosome will shift a little bit to the right to be able to accompany the last part of this, the last codon, the last tRNA that needs to bind. So shift a little. And now this one doesn't fit anymore, so it's going to fall off. And now, last one, U codes for A codes for U, A codes for U. So here it will come, and these three again will transfer to the last one. And then what you will have is all of those amino acids being transferred to this one. And so you will get this. And again, ribosome will shift one last time to see if there's going to be another part, but there isn't, so this one falls off. And now what it's going to do is it's going to reach a stop codon. A stop codon will basically, like in this case, it reached a stop codon, there's no more, so it's going to stop, it's going to break off, all the things are going to disassociate, and this chain will disassociate from the tRNA. So essentially, you'll have this left, your final product, the protein. Now this ribosome can go now and wait in the cytoplasm for another new codon to come and to make another protein that will be useful for something else. So as you can see, it really isn't so complicated. If you just follow the idea, it's not so bad. One last thing here. So this thing is called a protein, having a function in the body to affect something. And the bonds by which these amino acids are linked are called peptide bonds. Okay, just so you know, peptide bonds hold these amino acids together. So, just to reiterate, translation translates this paper into a protein using ribosome and tRNA, just like Bob the Builder needed this paper, his tools and his materials, he could finally piece them together to make this functional thing that's going to have a purpose. Just the same way your cell makes this protein. So I hope you understood this. I went slow just to make sure you guys can understand this. And now I will make some questions based on a transcript. I will get some questions, past paper questions of IB, and explain them in the next video regarding transcription translation. So I make sure to go check those out.